And hello, everyone, and thank you for joining us for today's e-showcase, PFAS Analysis by EPA 1633, Building a Comprehensive Capability. I want to take a moment to thank our sponsor, Waters Corporation. And I have the pleasure of introducing our presenters today. First, we have Stuart Earle, Senior Principal Technical Support Specialist, Chelsea Plummer, Senior Product Marketing Manager, and Jennifer Brzezinski, Field Service Applications Team Leader. And without further ado, I will hand it to our presenters.
Hi, everyone. My name is Stuart Early. Thank you for the introduction. Before we get started, I just wanted to give a background, a little bit about Waters at a glance. We are a liquid chromatography mass spectrometry company and also thermal analysis innovator. We have over 7,900 employees in 35 nations throughout the world. And we have in 2023 revenue of over $2.9 billion. We are one of the industry leaders in service and informatics, and we partner with our customers around the world to help ensure things such as purity of our food and water, efficacy and safety and quality of medicines and vaccines, as well as the durability of products we use every day. So we are in a wide market range, including academics, I should add as well. So next slide. So today we're going to be covering getting started with PFAS testing and kind of what you'll need to know for a comprehensive testing capability. The first part I'm going to do is the spotlight on the LCMS technology. My colleagues, Chelsea and Jen, will be covering the last two bullets for today. So next slide, please. Why are we here? Well, EPA method 1633, among many of the EPA methods for PFAS. This one came out in January and was finalized in January of 2024 for over 40 PFAS compounds in a wide variety of matrices, from wastewater to surface water, groundwater, soil, biosolids, sediment, and landfill leachates, as well as fish tissue. It was created by the U.S. EPA Office of Water in partnership with the U.S. DoD Strategic Environmental Research and Development Program. And it was validated, interestingly enough, if you look at the method in a multi-lab study, and it was a quite extensive and lengthy time for the development of this. And it's mainly meant to be used for Clean Water Act compliance, Superfund sites, and remediation of investigation programs, including DoD installations throughout the United States and world. Next slide, please. So just to give you a brief introduction to LCMS technology, or tandem mass spectrometry. If we look on the left-hand side here, you have the HPLC. You have a solvent, you have pump that delivers the liquid, an injector, and then the magic happens, so to speak, in the column. All right, and if you look there, the gray tube in there that says HPLC column, that's where the separation occurs. Next slide. You can see in this case, if we were doing some food colors, you'd separate into the bands, and then they're detected some way. In this case, a UV detector, or it could be a mass spectrometer, refractive index, or even fluorescence is used. And you can see the chromatogram there on the window in the computer there where it separates those components. Now that separation is done in the column with the packing material, the magic white powder, if you want to call it that. On the right-hand side, we kind of have a view inside the column where you can see as the analyte comes down, it's going to interact with either the mobile phase or the stationary phase. And depending upon that, its affinity for the stationary phase or the mobile phase will tell you the elution that it comes off, or when that band that comes off on the separation is seen. So as you're injecting, in this case, these were food dyes. They could be PFAS compounds out of a biosolid or a wastewater extract. Those interactions with the stationary phase and the mobile phase will tell you when it comes off. So things that have a stronger affinity for the column come off later. Things that don't like hanging around with the column material come off with the mobile phase quicker. So that's how the separation is done. Next slide, please. So now we get to the point of now that the sample introduction that you see down there, that comes from the column into the mass spectrometer. So the mass spectrometer has an ion source. in which you ionize the compound. Typically for PFAS, it's electrospray in the negative ionization mode. So we're looking for the loss of a proton or an M-H ion typically in PFAS analysis. Then to the mass analyzer where the ions are sorted and detected, and then sent to the detector and to the data system, and you get your results. Here, how much of each of my components do I have? Next slide. The sample introduction and ion source is shown here. This is a Z spray. You can see the spray coming across the top there, and then the yellow kind of Z shape there is how the ions get drawn into the mass spectrometer, and you're basically taking it from a solvent phase into a gas phase and ionizing it into the instrument. Next slide, please. Looking here at its, LC-MS technology or tandem mass spectrometer has been selected by regulators for PFAS analysis, mainly because of its selectivity and sensitivity. It's the gold standard, so to speak, in industry. It's used not just in environmental, in drugs of abuse and other assays. It's the gold standard. If you look on the right-hand side there, the picture there is of the instrument. You can see it's a benchtop instrument. The absolute is the actual larger instrument on the right there. On the left is the inlet, but it's a benchtop instrument. Why was it chosen? The high selectivity comes because it can reduce or eliminate matrix interferences by being very selective in the ions that go into the mass spectrometer. And it has a high sensitivity because you can get to very low levels with it. One of the other positive things about tandems is it's very good in quantitative accuracy and reproducibility, as well as robustness. That Z spray that I showed you on the previous slide is one of the things that we have on our system that helps with the robustness, because that cone can be easily replaced and cleaned without breaking vacuum, without recalibrating, and allows you to have maximum uptime when you're running a system. The typical analysis method is what's called a multiple reaction monitoring, or MRM. What happens is you have the MS1, the purple quad that you see there, which allows only a certain ion of a mass to go into the T-wave collision cell, where it's then fragmented, and then the MS2 quad allows only the ion that's fragmented to come in, or multiple fragment ions. So in many cases with PFAS, we do multiple ions from a single component, say Gen X, and then you can get multiple fragment ions from it. And that's the selectivity. That's really the power that you see here with the instrument is because of that. Next slide, please. So what does that give you? So you can separate. So the column gives you the separation power. The mass spec gives you the selectivity and sensitivity. And you can extract, in this case, you're seeing here carboxylate and sulfonates of PFAS, all right? Those can all be done in a single run. We've simply just extracted them out here to show you that you can easily look at them. And because of that selectivity, because these compounds all have different masses, you can also look at specific ions for them. In addition, because of the separating power of that column, you can also get things like you see there on the C6 and C8, the circled in orange there, the branched isomers. So not only the long chain PFAS, but also the branched isomers you can easily detect with this instrument, and you're able to give quantitative data for all of that. Next slide. So here's the full picture. You bring all of those together in a single run, EPA method 1633 or the ASTM method port as well, 8421, gives you 46 native PFAS compounds. You also have the internal standards that are added for quantitative reasons and recovery reasons. And if we go to the next slide, please. This is just an example. These are some spider plots showing you some results using a certified reference material for validation. You see on the left the PFAS in wastewater, and on the right the PFAS in soil. What you're looking at is the compounds are kind of in the sphere or the circle, and basically the certified value, the value that was in the material is in blue, the experimental value is in gray, and you can see the certified levels. And basically the take-home message is we had a trueness value of 92% for the wastewater, 97% for the soil, with the range of shown there of 73 to 112 and 85 to 120, well within the method requirements. So, and very low levels. Next slide, please. In addition, this same system can also be used to look at other EPA methods that are out there, such as 537.1 and EPA method 533. All of these can give you identification of the various PFASs required in those as well. Same system, just different column chemistries. Um, and keep in mind, though, the amounts we're looking at are typically nanograms per liter, parts per trillion. So kind of as a final thought here, um, what is parts per trillion? We all hear these numbers thrown out there, so I kind of put together a nice slide here. Next slide, please. You know, putting it into perspective, a nanogram of compound per liter, part per trillion, is 1 part per 10 to the 12th parts, or 1 drop of water in 20 Olympic-sized swimming pools. How appropriate with the Olympics coming up. 1 second of time in approximately 31,700 years, or 6 inches out of the distance between the average of the sun to the earth, which is 93 million miles. So just kind of a put in perspective the kind of levels you're looking at here and what you're trying to detect. And these instruments are able to detect at these very low levels. And then they even go further. They're even trying to go further down than that. So next slide, please. So I've kind of talked about the instrument, but testing for PFAS requires a complete comprehensive solution, doing the sample prep. So, well, first of all, collecting the sample prep, the instruments, the data collection and output, getting the data, either sending it to my LIMS system, getting my data, that kind of thing. Then also doing certified reference materials and some type of proficiency testing showing that I can do that analysis and I can do it reproducibly and reliably. So it's the whole piece of this and making sure that you can bring all of this together that's required. So next slide, please. So with that, I'm going to kind of hand it off to my wonderful colleague, Chelsea, who will be coming on to talk about sample prep. So take it away, Chelsea.
Thank you, Stuart. You can move to the next slide. So Stuart did a really nice job introducing the instruments component of why we use these for PFAS analysis. But as he just showed, in the full comprehensive solution, there's more than just the instrumentation to take into consideration for the end-to-end workflow. So let's get a little bit into why we use a technique called solid phase extraction for PFAS analysis. And there are a lot of reasons why we use it. I'm going to break down a few of these here. The first one is kind of because we're required to. So they're in all of the US methods for drinking water and now in 1633 for non-potable water as well as solids require a use of solid phase extraction cleanup steps in those methods. And these are also really important because they can help remove some additional interferences from samples, which helps when we look lab to lab in terms of the reproducibility of the methods themselves. So for 1633, for example, there was a large round robin study showing interlab reproducibility for setting that method up, and it required use of sample preparation. As I mentioned, kind of alluded to in that last piece, we're dealing with complex matrices in EPA 1633. We're not just looking at drinking water anymore. We're looking at wastewater. We're looking at groundwater, different surface water, sludge, soil, sediment, fish tissue. These are very complex sample matrices, and they have a wide variety of different components in them. When we're trying to get down to these nanogram per liter limits that Stuart mentioned really well in one of his previous slides, we have to think about the other interferences that may be coming from them and may be interfering when we're trying to get down to these low limits. So being able to clean up adequately is very important to be able to get at the limits of detection required for these methods. And also, we do solid phase extraction for selectivity in PFAS themselves. We, in most all methods, use a wax or a weak anion exchange sorbent, which has some selectivity for PFAS that we'll talk about in a couple of slides. You can move to the next slide. So let's jump into the sample preparation for 1633 in a little more detail. As I mentioned, EPA 1633 does require solid phase extraction. It uses both the weak anion exchange SPE cartridge and also requires an additional cleanup with carbon as well. So this can either be in the form of loose carbon or graphitized carbon black, GCB, which you'll see I'll refer to as GCB as we continue on. And depending on what sample type that you're evaluating for 1633, whether it's an aqueous sample type or it's a solid sample type, the cleanup steps that you go through have a particular order. So if you're working with a non-potable water source, first you do cleanup with a wax cartridge, followed by the carbon cleanup. And it's actually the other way around if you're working with solid samples. So if you're working with soils, biosolids, and sediments, you would use a carbon cleanup first, followed by a wax cartridge. But EPA 1633 is a little bit unique. Unlike some of the other methods that are very regimented and how you have to perform the steps in a particular order and the exact configurations of different solid phase extraction devices, EPA 1633 is performance-based, so as long as you're able to meet the method requirements in terms of the recoveries and RSDs of the method, it allows for some alternate modifications. And one of the ways that you can do 1633 is by using cartridges that actually combine the wax and the GCB sorbent into one device itself, which helps in terms of just ease of use. You don't have to weigh out loose carbon. It's a little bit faster. And we'll talk a little bit about that in a couple of slides too. You can move to the next slide. So I've mentioned that for 1633, we use wax and we use GCB, but let's get a little bit into why those two sorbents are selected. I hinted a little bit about wax having selectivity for PFAS. If we take a look at the upper left part of this slide, you can kind of see the breakdown of what a typical weak anion exchange sorbent looks like. It has different sites of retention. So we have a reverse phase retention backbone, as you can kind of see with our rings and our carbon chains there. But it's also been functionalized to have a weak base on it as well. And the weak base allows for ion exchange retention. Most PFAS are going to be some form of acid, so they're going to have interactions, charge, negative to positive, with this weakly basic sorbent. And we can manipulate the pH in our SPE method to allow for a selective ion exchange interaction between the acidic PFAS and the weakly basic sorbent. So we first start and we prep our samples and condition our cartridge to being at a low pH, which activates the ion exchange sites on the weakly basic sorbent itself and makes it have a positive charge. Therefore, you have a negatively charged PFAS that's going to have this ion-ion interaction. Then we end up rinsing the cartridge with a solvent, which is going to remove any neutrals that aren't undergoing that ion-ion bond there. And then we can manipulate the pH further by increasing the pH, effectively turning off and neutralizing the wax sorbent to selectively release the acidic PFAS. So you can see that that's the reason why most PFAS method that you look at are going to be using some form of weak anion exchange sorbent. In 1633, there's also the addition to add graphitized carbon black to the method. This actually allows for the removal of even more matrix interferences than wax alone. And if we think about moving into 1633 in these complex matrix, you can see where that might be effective. Also, too, we have some benefit with GCB in being able to better retain neutral PFAS. As I mentioned, the wax is really beneficial when we're thinking about those acidic sites on the PFAS itself, even though they also undergo the reverse phase retention, but it gives us a little extra push for those neutral PFAS. You can move to the next slide. And I'm not going to break this down in complete detail. A lot of it is really familiar to what I just talked about a little bit on the previous slide in terms of the workflow for 1633 SPE with pH manipulation, working through the cartridge. Over here on the right side, you can kind of see what a setup would look like on a vacuum manifold with the cartridges for doing your PFAS sample preparation. But if you want to see in a little bit more detail, I really encourage you to check out the link off of this QR code below. We recently just did an educational video where we filmed end-to-end a typical 1633 PFAS workflow using a vacuum manifold that you might want to check out for yourself. You can move to the next slide. And finally, too, I mentioned earlier that 1633 is a performance-based method, so it allows for some modifications that you wouldn't have to just use the loose carbon like I mentioned before. If you've ever worked with loose carbon before, you know it goes all up into the air. You may need to use some kind of dosing gun to really measure out the amounts that are required in the method. So you can really make things a little bit easier on yourself using a dual phase or bilayer cartridge. This is just some work that we did back earlier this year, showing that for EPA 1633, using dual phase cartridges with both the carbon and the wax sorbent for both aqueous sample types and solid sample types, that we are able to meet the recoveries of the methods using this alternate bilayer cartridge in both matrix types, well within the requirements of 1633. On the right-hand side are a couple of bar graphs where the gray bar is the maximum allowable recovery in 1633, and the orange bar is the minimum allowable recovery, and our recoveries that we observed for the average recoveries are in blue. And you can see that both for aqueous and for soil samples, we also did tissue too, but I, for time constraints, did not put on this slide, able to well exceed the minimum requirements of 1633, making this able to be slotted into this performance-based method. You can move to the next slide. And finally, as I mentioned, there's a lot of things to take into consideration. Sample preparation is a really critical part of doing EPA 1633 or any PFAS analysis. And we're looking at ways in our research labs to make 1633 even easier to run. And some of this involves using these new dual phase bilayer cartridges that I mentioned, just to make the analysis even easier and not having to deal with manual steps relating to loose carbon, but also using systems like the Promochrome SPE03, which allows for full automation of 1633, as well as other PFAS methods too. We're also able to use the instrument Stuart introduced, the Acuity Premier with the Xevo TQ Absolute, as it really pushes the limits of sensitivity that we're able to get out of a tandem mass spectrometer to even reduce the sample size, which effectively decreases the amount of sample preparation time that we have working with large volume samples. So there are a lot of things to look forward to thinking about ways to make this method even easier to run in the laboratory. You can move to the next slide. And with that, I'm going to hand it over to my colleague, Jen, who's going to be covering some of the training and support aspects for PFAS. Thank you.
Thank you, Chelsea and Stuart, and welcome everyone. If you could please go to the next slide. So I wanted to cover today the training and support as foundations for success for water soil analysis testing. So why would you actually need training and education? For those of you that have gone to the EPA website, you can download the literature for 1633, for 537. But for those of you that have read it, it can be very overwhelming. There's a lot of acronyms. There are quite a few pages. There's up to 70 pages. And there's really a lot of translation that has to happen from the end user. The documents are meant to be very broad so that the user can go in and then put their flexibility and their ability to run across different vendors, across different applications. So what we would like to do is help take out some of that flexibility and actually be a little bit more specific to help with the translation, to help decode the PFAS literature for you. So if you go to the next slide, please. What is not in the literature that you can download from the EPA websites are method specifics for the instrument, method specifics for the LC, for the mass spec. How do you run the software? How do you calculate? Okay, so many of those things are unique and new to people in this environment that are starting labs for PFAS testing. So what Waters would like to do is partner with the end user to help you be more specific, to actually provide documents and methods to you as users on how to get up and running and help decode some of the information that is in the EPA documents. So if you can go to the next slide, please. Okay, so Waters has a PFAS application user training solution for 1633, as well as for 537 and 533. The end goal of the application solution is to really walk you as the user through the prep, through the standard setup, through the instrument and method setup, and also train you on the LCMS as well as the application itself. Okay, once that happens, we will continue to provide not only on-site support for you and your colleagues, but we'll be able to help you remotely as you continue to implement the method in your labs. We'll be able to follow up, see how the chromatography is looking, and provide guidance for you and your team as you continue to implement this into your lab. If you can go to the next slide, please. So what does the training, the PFAS application user training solution provide for you? There are 2 guides that we will provide to you as a customer and as a user. There is one guide that will help you walk through the PFAS solution setup. So your standards, what standards to buy, how to make them, how to dilute them, how to prepare them, what solvents to use, and maybe some other lab consumables that are important. We will walk you through the best practices on how to actually prepare your solutions. The PFAS Application Success Guide will walk you through a lot of the software portion, okay? Once you start running, you have quantitation, you're going to be calculating. You want to be able to generate reports for the end user. You want to be able to provide information to LIMS. So we will detail and walk you through how best to do that to help with your success in the implementation of 1633 or some of the other applications as well. If you can go to the next slide. So just a quick recap, okay? We will provide solution setup and the success guide. We'll also restore LC methods, MS methods, processing methods. A lot of the calculations that are outlined in the 1633 guides, but not detailed. We'll actually just restore those for you on your workstation, so it can eliminate a lot of the development that you might have had to work through, and we'll be able to get up and running much quicker. We'll be able to, again, provide expert guidance on site or virtually, and your team will receive a certificate for the training, so that you are, if you're in a lab that needs training records, we'll be able to provide a certificate for you that you attended the LCMS training, and specifically for the PFAS application. So our goal is to help you save time and effort that is needed during the development phase, and really focus on the output and the actual testing. Next slide. I want to thank everyone, and does anyone have any questions?
Just a reminder, go ahead and put your questions in the chat module. And we actually do have one question that has already been submitted. If sample pretreatment is required before SPE, what filter material is recommended to minimize sample contamination with PFAS leaching from filters? We are currently using nylon, but are interested in your experience.
I can comment to some of that, but if Jen and Stuart want to jump in to being more laboratory based, I'd be glad to take your advice too. Typically, the filters that we end up using are the the Acrodisc syringe filters. Those are typically the ones that we recommend using, but I can ask my colleagues if they have any other suggestion.
Yes, I would agree with Chelsea. The Acrodisc is definitely one of the filters that we recommend. You'll also note that Waters and some other vendors will also tell you that their discs or consumables are PFAS free. So that's something to also think about, and we will guide you on some of the pitfalls that we've seen and some of the things to think about going forward.
I can add one more thing too here. For some of it, if you have samples that have floaties, is that the right word I want to call it? Some of the EPA methodology recommends using glass wool or something in the device above, but one trick is to rinse it with LCMS grade methanol and clean it before you use it. So, and then you're still going to have to use that as well on a blank, because while everything can be PFAS free, PFAS low, the challenge is all the cases is seeing what the levels are and making sure that the PFAS contamination in your lab is at a consistent level and something that's manageable. So
All right, perfect. And so, what type of tubing is recommended for automated sampling in wastewater? Are there interferences or contamination with the use of certain sampling tubing?
So in
avoid
our
Teflon.
systems, I was going to say I can talk a little, and then you can, I know I'm kind of taking. What we recommend on our systems for LCMS for PFAS, we have a full kit that we outfit the LC with to replace any of the tubing that essentially to remove anything that would have like Teflon to it, so that you have significantly reduced anything that would be coming from the system. Besides just changing the tubing as well, if you're running an LCMS for PFAS analysis, what you'd want to have in place is also using an isolator or a delay column. Even as much as we change in terms of the tubing or anything in the system, as Stuart was mentioning, even if you're using the highest grade of solvents, there's always going to be some degree of contamination that you have to deal with. And using a delay or isolator column helps move that away from your analytical peaks to reduce that interference that would be going into your method.
I agree. That's the biggest thing as well. And if you're curious to see what it looks like in that, on the slides that you'll have available at the end of this will be a link. You can go to our waters.com/PFAS site and see some examples. It'll show you quite nicely. So
Okay, perfect. Is training and technical support included when leasing an instrument, if such option is available?
leasing is available and the training and support can be included in the pricing of the leasing. Yes.
Are you supporting any LCMS, MS, or some specific model manufacturer?
Obviously Waters. um, any of our tandems. Depending on it really is going to be driven by which method you want to do and how low you need to go without sounding like a limbo concert. But we have a wide range of tandems. So depending upon the level of the EPA methodology you need to use, our sales reps will guide you through which is the best instrument to get you the performance that you need.
Great. Do you have any suggestions for mitigating PFAS contamination from the laboratory environment?
That's a great question. We actually have a guide on our website called Mitigating PFAS Contamination in Your Lab. So there is a lot of stuff. So I would urge you to go to that and take a look at it. So I would defer to the very large white paper that we put together on the water site that goes through that.
There are so many different things that
Yes.
can contribute to the contamination, from if you're wearing specific type of makeup to clothing to using aluminum foil to cleaners that are happening in the floors of the lab and everything.
And
There are so many
and
considerations to have to take.
yeah,
We
hand
could talk about that a
creams,
whole hour.
yeah, lab coats, everything.
And
So
I think a little birdie just told me that the mitigating contamination guide is linked in the slides too.
It is.
Okay, perfect. All right. Is the degasser a source of contamination? Can it be removed?
Most of it can be a source. That's why we have the isolator column. So the isolator column delays it coming off. So as Chelsea mentioned, it is after the pump and before the injector. So you're delaying. And most of our degassers now, the Teflon in it is pretty much minimized. But that's the whole point of the isolator column.
And someone is requesting a link for the lab contamination list, but that'll
That'll be in the
located.
slides. Yep.
That's going to be
There'll
in the
be
slide deck. Yeah.
there'll be several extra slides that you didn't see today that have links that you can click on to specifically look at different things.
Yes. And again, the slides will be available tomorrow. You will receive an email that will have a link that will take you back to the landing page. There will be a link to the slide deck, and everyone will have access to that. Have you started to look into procedures for PFAS in air?
Yes, we have. So we don't sell the air handlers or air samplers, but once you sample it onto the filter, yes, there are methods that are out there that we've investigated for that. So
We've also started to look into using the system Stuart described with the mass spec with an atmospheric pressure GC source to look at more volatile PFAS that aren't as amenable to liquid chromatography.
Okay. And what is the recommended method for drinking water samples?
It depends on
yeah.
what you're trying to run in your lab, but it could be EPA 537.1 or EPA 533. Those are the two major ones, but there are other methods that exist as well.
Okay. Stuart,
That's gonna probably be driven by regulatory. What's your
Yeah.
regulatory requirement in a particular state, county, or if you're outside the United States, country?
That's exactly right. Or if you're doing more research-based investigative work, you may even add. We work with companies all the time that kind of have their own lists where they've done EPA 533 modified and add additional things to theirs, depending on, like Stuart said, if it's really purely regulatory or otherwise.
and what tools are used to conduct DNA for drinking water?
Not sure what the question is asking.
Yeah, I'm not sure either.
So Harrison, if you would want to just restate your question down there below, see if we can kind of understand it a little bit better. And while he's doing that, have you extracted and analyzed plant samples?
Yes, we have. That is a whole separate piece on you'll get a link to the site. There's been a lot of work done looking at it in food, feeds. Um, Unfortunately, containers, cosmetics, you name it, someone's looked at it. So yes, there are methods.
We do have a pretty recent application note where we've gone through in fruits, vegetables, and baby food samples using essentially the 1633 methodology end to end on those kind of samples that you may find interesting too.
Okay. And do you have a method for incinerator stack testing and short chain fluoride?
Short chain fluoride, yes. There's a couple application notes that are linked in the slides that will take you to some work we've done with looking at short chain. The challenge with short chain is going to be the fact that some of the short chain compounds, such as trifluoroacetic acid, is everywhere in the lab. So sometimes the lab contamination can be more the challenge than actually detecting it. But yes, there's actually a nice application note on some of the short chain PFAS compounds.
Okay. And so Harrison has resubmitted his question. Let's see if it's a little bit more clear. Do we have a simplified tool for running DNA for drinking water?
That's beyond the scope of what this talk was all going to be
Yeah, I
about, which
don't.
was PFAS and environmental samples. So, I mean, if you're looking, DNA is kind of a general term, so I don't know if we can really answer that. You might want to reach out. You can analyze DNA. I'm not sure what you're defining as DNA, whether it's animal, human based, or whatever. So that's kind of a, I'm not sure.
We've got just a few more minutes. If you have a burning question, go ahead and change that to option to everyone and go ahead and submit a question to us. And Harrison goes on to state for analyzing drinking water quality. I'm not sure if that.
If you're asking about different compounds of DNA, we can analyze DNA. There are methods that are out there. I've not seen any that have ever been really applied to drinking water. It's been more in the bioanalysis world, but yes, there are methods for analyzing DNA, and our instruments can be used for that. So that is, I would say, go to our waters.com website and search for oligo or
Yeah,
DNA.
I would agree. It'd be kind of what I believe is being asked is you doing almost like an omics-based targeted workflow with LC-MS, which you could do. We would probably encourage you to reach out to somebody else from Waters, and that we can connect you with those kind of people if you reach out after and have some further discussions on that.
All right. So we've got a couple more questions that'll probably take us to the end of our presentation today. We have, will sodium hypochlorite affect the PFAS sample? We use sodium hypochlorite in our wastewater disinfection process.
It should not, because PFAS doesn't degrade from that. So your PFAS samples will still be there. It likely will go through the cartridge.
All right. And Stuart looks like he may have responded to this next question. Do you have handy information? What is your blank versus MDL versus log values?
That would be on the application note, that information on our MDL stuff and our versus blanks. That was kind of in that spider plot that I had there, but the application notes that are linked at the end of the presentation also include some of that information.
All right. I think that may sum up our questions. We've got about 1 minute before we'll close out. And if any of our panelists would like to make any closing comments.
No, I just appreciate everyone sticking around for 45 minutes to hear us talk. And certainly, hopefully, this has given you at least some knowledge to be dangerous. It's not, certainly was not all inclusive, but hopefully the information you'll get in the PDF and the guides can take you to the next step. And certainly, if you have further questions, feel free to reach out to us or anyone at Waters. We'll be happy to help you and guide you, and at least, you know, in the journey, is that the right word I want to call it, of PFAS analysis. So anyway, thank you everyone for coming.
Thank you.
Absolutely. And we would also like to thank you, and we certainly want to thank our sponsor, Waters, for today's presentation. Again, if I have not stated before, the recording of this webcast with audio, visuals, and a link to the PDF of the slides will be posted on the landing page. You all will receive an email tomorrow that will have that link in there. It'll take you right to the recording. So thank you, everyone. Thank you to our panelists. This has been great. This concludes today's eShowcase. Thank you all for attending. The recording will be available at webfiresguide.web.org. We will send all registrants an email tomorrow with that link. And please visit our event calendar to sign up for future webcasts. Thank you, everyone.
Thank you.
Thank you.
Thank you.